Good morning, dear students. Today I will talking about the DNA extraction. We have some notes about them, maybe in the last year, or maybe I talking about them from the last lectures. Here, for start DNA extraction, you must have some steps or some stage to done this extraction. You must have the sample who you want to take the DNA from it. These samples may be blood, a plant, animal tissue, any other things, especially from the living organisms. So that these steps, the first one, a sample, and you must have buffers and reagents for interaction, steps for doing that, and storage. This DNA, you extract it, and downstream application. This is meaning where you used this DNA, for what the purpose of extracting the DNA, for what the purpose for this. Now, DNA extraction is a process that it is used to separate the DNA from blood, animal tissue, plant tissue, or other living components by using chemical or physical methods for biological samples. The purpose of DNA extraction. Purified DNA will be used in the future, in further investigations, such as, here, when you purify the DNA, this DNA will be used in the PCR or in the DNA sequencing in the NCBI for the gene bank, or maybe in other treatments like Southern blotting, and also you can use it for the gel electrophoresis. Purified DNA will be used in further investigation, such as, I repeated that, such as DNA, PCR, DNA sequencing, Southern blotting, and electrophoresis or gel electrophoresis. And here you can see by my pointer here the cells, you put it, and this is the reagents and chemicals for extract the DNA. The DNA appear like cotton if you see them, or if you saw them before. The genome. Sometimes we have the term, the genome. Genome, it is all genetic materials. All genetic materials in an organism. All the DNA in the organisms called the genome. This DNA, most of them is in the nucleus, and some of them in the mitochondria. For the viruses, they don't have DNA, they have RNA, or almost of them have RNA. DNA consists of double helix, or the DNA as a double helix on the chromosomes. The chromosomes inside the nucleus. What is DNA, or what's the meaning of deoxyribonucleic acid? DNA is a nucleic acid made from carbon, hydrogen, oxygen, nitrogen, and phosphorus. A fundamental molecular found in all living things or in all living organisms. We can find the nucleotide in all of them. Carries the genetic information in the cell so that the DNA holds all the inherited information about the organism. Most of the DNA is found in the cell nucleus and some of them in the mitochondria, capable of replicating and synthesize RNA, so that the DNA by messenger RNA may be changed to the RNA. Contains instruction for our body cells to perform their specific functions, so that the instruction on the DNA will lead to perform the specific function, like made protein, or use them to doing all the process in the body. Here we can see DNA can be extracted from any cellular tissue, like this is bone marrow, or the bone. This is the bone. And inside them, the bone marrow. Also from the skin from animal or human, anyone. The blood also, you can take the DNA from it. Saliva, also we can take the DNA. A plant and bacteria also, you can take the DNA from it. Sometimes the viruses, you can take the RNA. In any way, the buffer and reagents used in this process, in the DNA extraction, you must need a trace HCl solution, PDTA solution, an HCl solution, sodium dodecyl sulfate, this is an important reagent, a proteinase K, that is enzyme, phenol, a chloroform isomide alcohol, isopropanol, and ethanol. Maybe we have some other things like the Eppendorf, you need the vortex, need some tubes. This is, you can find them in your lab during the DNA extraction. Some buffers. Buffers used to maintenance the solution or the maintenance the DNA. So that some buffers and solution in DNA extraction, like SDS, used to aid in lysis cell during DNA extraction by removing lipid from cell membrane. The cell membrane, as you know, the structure of them, have a lipid, so that the HDS remove this lipid from the cell membrane. Proteinase K cleavage. Cleavage meaning analysis a glycoprotein and inactive or stopped working of the RNase and DNase. Inactive these two enzymes. Trace HCl EDTA buffer. The solution solubilized, meaning that make the DNA like solubilized DNA while protecting it from degradation, maintenance the DNA. EDTA gelating agents. Chloroform remove the aqueous or the water aqueous phase with the minimal cross contamination from the organic phase. Phenol to ensure a clear separation between the aqueous and organic phase after centrifuge. When we make the centrifuge, we have two layers, and between these two layers, we can find the phenol between them. Also, to continue them, NaCl, the rule of the salt is to neutralize the charge of the DNA's sugar phosphate backbone. This makes DNA less hydrophilic or less soluble in water. Isopropanol used to precipitate DNA from the solution. Ethanol is used to wash any or to wash away the DNA and remaining salt residue. That each of them have a special working to achieve the extraction DNA. Aqueous phase, this is related with the water, containing DNA and water. Organic phase, containing denatured proteins and other cell components. Must be used all these reagents and solutions and buffers when you want to extract the DNA. And each of them have a rule for these processes. There are three basic steps during extraction of the DNA. One of them, or the first one, cell lysis, using proteinase K and SDS to disrupt meaning stopped to disrupt the cell membrane and break the nucleus to release DNA. A precipitation, Na, this is coming from the NaCl, Na plus, because the DNA have negative charge, and to neutralize the DNA or this solution, we add Na, and you know the Na have positive charge. So that here, precipitation Na to neutralize the negative charge of DNA and make the DNA stable. Then alcohol, like ethanol or isopropanol, is added using the DNA to precipitate out of the aqueous solution because it is not soluble in alcohol. Purification, this is the last stage or last step. DNA is separated from other unwanted materials by using alcohol. Then a purified DNA is redissolved in water for easy handling and storage, and storage them in the very low Celsius, very low degree Celsius. Here, a procedure of DNA extraction, like for blood. We have some different procedures, and this is related with the kit of different companies who make them, and so that we have some differences in each kit. For the blood, we can use this type or these steps or extract the DNA from the blood. And this is, we cannot save them. You can put it in the bench and you doing each step and sequentially. Because it is very difficult to save them, so that you must be putting in your bench and take each step to do not make any mistakes. But we have some solutions like this is AW1 and AW2 and AE. This is important solutions. The company makes them to extract the DNA. So that I don't read all of this, you can apply them when you extract the DNA. Here, also the procedure of DNA extraction from blood. This is the steps, and you can follow these steps until you take the DNA, then store them at minus 20 Celsius. This is like easy shape to understand for going to each step. The procedure of DNA extraction from bacteria by boiling methods. We have two different types. If you extract the DNA from the plate, we can follow these structures. And we have another one, extract the DNA from a broth, liquid culture. This is the second one. For the first one, for the extract or extraction the DNA from a plate, suspend 4 to 5 colonies of bacteria in 300 microliter of sterilized nuclease-free DDH2. This is meaning deionized water, or the waters used in the molecular biology called DDH2O. Second, incubate the suspended bacteria at 100 Celsius for 15 minutes. Chill, lower the temperature, meaning here, chill the suspend samples for 5 minutes, put in the freezer. Centrifuge the samples for 10 minutes at 15,000 RPM. Then store the supernatant, and this is contain the DNA at minus 20 Celsius. For the other type, or the second type, extract DNA from broth like liquid lectures. Transfer 1.5 ml of bacterial broth into 1.5 ml microcentrifuge tube. Centrifuge the sample for 10 minutes at 15,000 RPM. Discard the supernatant. Resuspend the pellet, the galls, meaning here, with 300 microliter of sterilized nucleus-free DDH2O, or deionized water. Incubate the resuspended bacteria at 100 Celsius for 15 minutes. Chill the low of temperature for the suspended sample for 5 minutes by putting them in the freezer for 5 minutes. And here, centrifuge the resuspended sample for 10 minutes at 15,000 RPM. Then store the supernatant, which contain the DNA at minus 20 Celsius. Storage of isolated DNA. The original stock of DNA will be labeled and stored at minus 80 Celsius. And maybe we don't have these equipment for the 18 Celsius. We can put them in the minus 20 Celsius. The working DNA sample will be labeled and stored at 4 Celsius or at minus 20 Celsius. And for 4, when you used it directly. But if you want to working after that, about more than 1 day, you can put them in the minus 20 Celsius. Here, when you extract the DNA and you get the DNA, when or where you used them, where you used this DNA, downstream applications, meaning where you applied this DNA. Forensics, this is in the medical lines. You can, when anyone have, or when you have the case of a criminal, you can use the DNA for the fixed the criminal. Also, the second one, paternity tests. This is meaning, how do you know this person, it's belonged these two parents from father and mother. You can doing that by this tests. Third, ancestry tracking or phylogenetic tree, meaning to know the DNA when you obtain it. It must be nearly for each of the organisms. This is doing that, or this phylogenetic tree, meaning the related between organisms, and the gene bank and NCBI have advanced programs when you used this DNA or you sent the sequence of DNA to them. Medical tests, also you can use the DNA for them, and like investigation in the mutation, like genetic engineering, sometimes use the DNA to improve, especially for the plant, improve like tomato, like potato, any other plants. Vaccines, you can use them also and apply them and obtain the results of these vaccines. Also use them as hormones or investigating for the hormones. This is briefly the pre-lab of your work, and you know that we cannot apply them here by online, and I wish to come back soonly to the campus, and I want to apply at least the extraction DNA and using the PCR, so that we cannot apply all things and in the online. In any way, thank you for listening. And if the Zoom cutting, I will make another one for the discussions and for the take your names presented here in these lectures. Here, I will stop the recording and to take your name. Now you are 10 participants here. 2 of them are absent. Juan.
بلى دكتور.
Tanya. ديانا.
نعم دكتور.
دانيا وينها؟
ما أدري.
ها؟
مو موجودة من البداية.
زردشت. جيمان.
بلى دكتور.
أنا دا أشوف أسماءكم، لكن هم مع ذلك هم دا يعني أطلب. زينب.
بله دکتر.
دیاره هین سرخطینین هر گوه که حتی ایک جوب داد.
بله دکتر، بس حتی الميكروفون تقارين بجاي كادي بيد.
سولار. بله. سولار. سولار سرخطینین. اوکی. لانا.
بله دکتر.
مریم.
دکتر عفو دکتر او زردشتی مسجك فريقر السرچك دنگه من اسنشن بكم ديرا مشكلة قد دنگي داي
بلا مشكلة كنت تنجي له هاي ويا غايب أجي. مريم. مريم. مريم غايبة. هلين. يا سرختي يا شوي كا شوي كات. يحيى.
بله دکتر.
ويسرى.
بله دکتر.
تمام. سپاس یابد کم هکه و بسیار کبیت شده کبیت هنشن آنلاین جیبون برای که هنشن زی سر اوه بمیزن سر
دکتور ببوره مای کامن ونت بی خودین زانم بو
کیش که اوه کیا
زردشت دکتور
زردش نو ویتا ید سر بس کنه کتل کیره بود
خودی دکتور هستی دو بیم بس ونت بیم ریسوله جی فرای کرد
چنینه قدیمه
ببوره
عفو دكتور او فقير هاليني جري سلا فريقر يوج سرچك دنگه من نايدن
کیش که
هاليني
هلين من هوني بيجي حاضر هلين مديت
دکتر بسیاره که
مريم
پروسی
مريم وسولار وديانا سكت حوزرين بلي كلام كده
دكتور داني ما موجودة، أنا ديانا موجودة.
ديانا موجودة أنت؟
إيه داني ما موجودة.
مني؟
دانيا.
دانيا موجودة.
دکتر بس عفو او دي محاضرة بروسيجر مطلوب منه؟
لا بروسيجري دي دنيا سرميزة ودي شولكي كاش برنوكت تمام كاش برنوكت كوكا
تمام سپاس.
بروسيجر دي دينا افتح حتى هكا ما شو لكيرجي وقتك الامتحان عملي ونهائي دي بروسيجري طبعا كمدون ما دفتر ودي تبخو بس من نتيجة فت السبوسيوات كما ترسم قطعة ببيتخات بس هم بظنك وما هو بس مدود غايبا سولار ومريم ودانيا هي أوك إشكالك السبوسيوات كم طبعا عملي فرق ديال نظري عملي موديل تشي كم زيادتر موديل تشي كاينة لا يفصون حتى أبجيد تمام هذا زردشتي أبو خويا نفيسي وقلتي از دنك نوهت تمام يعشت سبوسيوات كاين أمدي أبدي روبصينم هكا كسي تشو أوك نبيت يعني بسيار اكتبينيك اكستراكشن قالكو يعني ايديا يعني يوبصونو هي دي بينيتو وديل سيرفون خطواتوشي وقال جوروجي ديل سير خطواتوشي بس نتيجة دار نوكافيت يعني خلت يكو هاي مثلا تركيزو دو يعني هند جورا دي شولكي ما يوديتي دي شولكي هاي فكيت نتيجة ينويني هرو دي كرار كي بس نتيجة نايني خلت يتعرضكي دو تركيزكي دو وهند جوروجي هكي شولته هم يدرس بيتا ما أول جولي دار كافيت دبلا سپاسی هوت کم هکی هوت چه هوت نبید بسیارک و بخیر بچن حتی انشالله خود ریکه که وقت کرد و اف ام چین اف هکان بجین چین اف کمپاسی
زور ممنون دکتور
در چوا بخیراتن سرسری ما خیر بچن بیم الله